has a few interesting, I would say, ways to move forward beyond this simple model. I'm going to be using a phage system as a model system, and usually when I talk about biology, I take out my phone and use the phone a friend option, and I call up my biologist friend and collaborator, Holly Wickman. However, my phone doesn't really work in the UK, and it's quite early in the morning back home, so both of those things. Fortunately, we have Forrest here who knows way more about any of this phage stuff than I do, and so I'm going to give you a very, very toy version. We're talking about adaptive evolution, and since there's a lot of ecologists here, adapt does not mean you change your behavior to adapt to the new environment. It means that through mutation and natural selection, a new type emerges that is more fit than the previous type. Also, I'll mention, so I used to mention a lot of students, but this work is fairly mature, and so almost all of these students have now gone on to other positions, and so, well, one quit after a master's degree and ended up at Wall Street. So that's Craig Beisel at the end there, but and he's doing just fine, and Darren is at Florida State now, and Craig is still doing a research professorship here at the University of Idaho, and Holly Wickman is my main collaborator, and she is the one who designs and does all of the experiments for this. Again, we're talking about, so let me give you a little bit of an outline to begin with. I'll talk a little bit bravely about the experimental system. Then I'm going to talk about an fairly old paper now that appeared about 10, 12 years ago by Alan Orr that's based on some work by John Gillespie, who's a pretty famous population geneticist who worked in the 80s. And it's based on a very simple idea in retrospect, but kind of a very clever idea. And the idea is that, well, there's a lot of available mutations for which natural selection can act on. The ones that are adaptive would tend to be a small, relatively small pool among the larger set, and so they would be considered rare events. And there's a theory in mathematics about rare events called extreme value theory. And could extreme value theory sort of inform these idea of rare events in evolutionary biology? And so Alan Orr came up with a theory about that, and at the time, Darren Rakita, when he was a student, decided that he would use his phage system to try and actually either validate or refute that particular theory. And so it made several predictions based on extreme value theory about how adaptation would work. And as it turns out, did the theory work? Well, actually the answer would be in the form that Alan put forth of extreme value theory. The answer would be no. But for an expanded version of extreme value theory, the answer was effectively yes, it did fit the data. But you had to expand your view, and that was one of the things that I did, was write a theoretical paper sort of expanding on that, and Alan and I actually worked together on that. And that's a fairly old paper, came out in 2008 and barely got into genetics. It just had 2 more equations, and it would have been, they would have sent me away. So we're going to, but I did find out.
Where did you bury those 2 equations?
Bury those 2 equations? Yeah, that's right. It was, well, fortunately, I found out later that it was Sally Otto was the reviewer, and she's just the right person to push it through. So that worked out very well for us. And so we will talk about that theory of Orr's, which really talks about the first step of adaptation. Then I'm going to talk about an extension of that to looking at 2 steps of adaptation, and I'll explain what we mean by the first and second step of adaptation. And then we're going to look at the data versus this theory. And that'll get us to about 2010. And then one of the things that I like to point out is this extreme value theory approach to try to explain adaptation would leave a biologist maybe feeling a little bit underwhelmed, because while you use these extreme distributions, they have parameters in them, and it's hard to say what those parameters actually mean biologically. And so yes, you can fit them to this particular set of distributions, and you can say that you can make some predictions about how adaptation and evolution would go based on those predictions. But it would be nicer to actually have at least some view of what those parameters mean. And so we're going to attempt to do that using an old idea from Fisher, who developed this idea of what he calls the phenotype fitness map. Now, the data that I'm going to show you has no phenotypes in it. So phenotypes are traits, and for viruses, they have a few simple traits. They attach, they co-opt the cell, and then they lyse the cell, explode it, and they move on to their next victim. So we're not actually going to, but it would be interesting as a next step to actually start looking at the specific phenotypes in this phenotype to fitness mapping. But this is what Fisher came up with, this idea that adaptation works is in this big multidimensional space where as mutations arise, they move you around to new phenotypes, and those phenotypes map up to a fitness, and that fitness tells you where you'll go next. And one of the things that he did that I find very frustrating is he used a normal curve for his phenotype fitness map, which anybody who studies probability and statistics will find that very confusing, because he's using the normal curve not as a probability distribution like all of us do. He's using it as an actual physical map that takes the phenotype and assigns it a fitness. And why the normal? There's absolutely no reason except that it's a symmetric function. So I've done a little relaxation of that, which I think is at least on a first order reasonable. So I use quadratic, and it's just based on the fact that if you're sitting on the top of a hill and you have a smooth function, all functions are quadratic locally by Taylor's theorem. And so that's just sort of just like we're standing on Earth, it looks flat, but in fact, get up above, it's curved. And so it's just using local properties. And so I think, and actually everything that Fisher derived really works just as well for the quadratic as it does for the normal. You just need a peaked where you're peaked. And so what we're going to do is I'm going to reinterpret most of these results that I got from the extreme value theory using the phenotype fitness mapping. And the parameters will now have actually a meaning, which I'll tell you, and and which type of extreme value distribution you would expect to fit the data is now going to actually depend on how many phenotypes are affecting the trait. And we'll show you that it suggests that for the virus data, at least for the experiments, that maybe 2 or 3 phenotypes, 1 to 2 or 3 phenotypes are affecting it, and it can't be a large number of phenotypes or you get a totally different pattern. That's what it suggests, and of course it would need experimentation to go beyond that. Okay, now I've been using the word fitness, so I hope everybody understands what fitness means. So if you go to the gym regularly and you have a very strong body image, and you lift weights regularly, but you don't fail to leave any offspring, you're not fit. But if you're like my father, who drank 2 martinis a day and smoked a pack of cigarettes, but happened to have 12 children, he was very fit. So that's what we mean by fitness. It's based on reproductive success. And in the case of, there can be a debate on what is a fitness measure, but the fitness measures for phage are pretty easy. It's just growth rate. And you can actually do legitimate fitness assays and measure those growth rates. They're noisy, so you have to do them over and over again, but you have a very precise idea of what fitness is. And so now I'll explain. So the phage that Holly works on is a bacterial phage. The version that she calls is ID11, which comes from Idaho. And it has a very interesting geometry, which we're not going to take advantage of here. It's a very small phage, 5 kilobases total. She knows where all of the genes are. She knows most of all the proteins, so they understand this phage pretty well. The idea of how evolution works is this particular phage likes 33C, so we're going to challenge it with a higher temperature, so that is going to be the selective pressure to 37C. And effectively, you just put them, and you have lots of hosts, so we're not interested, we're trying to keep it simple, so there's lots of interesting questions about the host being bacteria. Bacteria is essentially what this particular phage uses, so we're going to be washing these out using new fresh bacteria at each flask. They grow up from we use different bottleneck sizes. One is a small bottleneck is 10 to the 4th, a large bottleneck is 10 to the 6th, and we replicate. And then at the end of each experiment, we do full genome sequencing, and we actually can track the adaptation. So we have our wild type, which has been sequenced. We sequence each time. If we see more than one mutation, we back up until we get the one that came first, and we can actually track to see as these mutations occur one at a time and are sweeping through the population. Now, a lot of dynamics. There's a lot of things you might wonder about. Is there clonal interference? Often, yes. Depends on the bottleneck size. Are there other complications to the dynamics? Yes, that's true. For the purposes of this talk, we're really mainly talking about the mutation that may not get all the way to fixation, but the one that starts to take over, and on that second background is where usually the second step of adaptation occurs. So it's a bit of a simplification of more complex dynamics, which are very interesting to model in themselves.
So, can I ask a question?
Yes.
So you say you sequence the genome. This is genome of the phage.
Genome of the phage.
But is this like a meta population of mutant phages at every cycle?
Well, you can get a pretty pure strain, and you can really basically see that it's exactly one base pair different from the previous one, and then one base pair different from the previous one, and you can actually, in this particular set of experiments, you can track the adaptation.
But do you sequence purified phage, or you just sequence all the phages at the end of every flask?
It depends. I think that sometimes, I can't remember how many passages they go through before they do their sequencing. Let's see, but I think they don't do it at every one, but if they end up seeing more than one mutation, then they'll go back. They keep the phage they keep samples at every single step. Okay. So as you can see that we're using there's the two different ones, the small bottleneck and the big bottleneck. As it turns out, with the bigger bottleneck, you are going to get more clonal interference. With the smaller one, it's going to take longer to evolve, but because the mathematical theory that Gillespie came up with was, he calls it swim conditions, strong selection, weak mutation. So as long as the selection is much stronger than the mutations, but the mutations have to have this restriction that n mu is less than 1. And so for phage, n can be pretty big. And so n mu often is not less than 1. And that n is an effective size that's really determined by the bottleneck. Okay, so for the smaller, so when you bottleneck down pretty severely, you can mimic these what Gillespie calls swim conditions a little bit easier than if you don't. Okay, and so ID stands for Idaho, and the very first paper that came out that basically looked at this theory was by Rikita in 2005, and it looked at some of the predictions of Gillespie and Orr's model. But we'll move on from what that said in a minute. And so this is just a little toy idea of how adaptation works. So here's your wild type, and you measure its fitness on that line, and a new mutation occurs, and let's suppose that the T changes to a G, and of course most mutations that arise, and in fact in phage, you can create conditions where every mutation there is has arisen somewhere because there's so many of them. Of course, most of them die out very quickly. So first one that dies out, second one, finally you get one that hits, and even if it is more fit, it's still not guaranteed to sweep. There's a certain probability that it'll sweep through. So if it hits, and if it sweeps through, then we say that's one step of adaptation. Okay, so here is what we call so here's the basic idea of the extreme value theory that Orr put together, and I'll tell you a little bit about the extensions. So the idea is that if you were to look at all of the available mutations, they'd be distributed according to some funny distribution, but out in the tail of that distribution would be the adaptive ones. And so if it tails off, like many distributions do, like a normal or a gamma, or this is the part that's a little confusing, the Weibull distribution, there's all kinds of them that just sort of tail off, and the extreme value theory says that if you're out in the tail of that distribution, if you measure their fitness relative to the wild type, it would look as if it were drawn from an exponential distribution. Okay, and that's this middle one here. If you expand extreme value theory, actually, and when you fit the data that Darren got to that exponential, it doesn't fit that pattern at all. So the basic pattern of an exponential distribution is you should see the most fit one should be on average twice as fit as the second most fit, a third, 3 times the third most fit, and so on. So then they pile up near the beginning, and there should be these sort of gaps. Now that's on average in a regular sample, but that's the kind of pattern that you should see. And if you see something away from that pattern, then that would suggest that the exponential is not correct. But extreme value theory actually has what are called 3 domains of attraction, and you can describe them by this distribution. And so there are 2 parameters that determine where you're at. The one that determines which domain of attraction is what we call this kappa parameter. And if kappa is less than 0, we're in the Weibull domain of attraction, which is very confusing because the Weibull distribution is not in the Weibull domain of attraction. So and I don't know why, but that's the name it's given. And basically these are truncated distributions. And the other is the other extreme is called the Frichet, where kappa is greater than 0, and these are what are called heavy-tailed distributions. So those are distributions, I don't know if you've seen the Cauchy distribution, and these heavy-tailed distributions are very strange when you simulate from them, because they look like just regular distributions, and all of a sudden you get this big observation way out there, and any experimentalist would say, well, what the heck? Something went wrong with the experiment and throw it out. And in fact, this model would actually predict that occasionally something crazy will happen. Fortunately, it doesn't appear that any of the data we have fit into that framework. But here's where things actually seem to happen in the Weibull distribution, where we have a truncated distribution. And it makes at least some sense that you would expect that if you're looking at adaptation, there might be a physical boundary by which you cannot get any more fit than that. And that's basically what it says. It just develops a series of probability distributions that are bounded. They have to have a certain property to them, in order to be an extreme value distribution, when you take and shift out and look at the shape of the distribution out beyond the shifted point, it should have the same shape as the original one. And that happens for all extreme distributions. For the exponential, you have to shift out and then rescale. For the exponential, you get the exact same distribution every time. You shift and rescale, you get the exact same distribution. For all the others, when you shift and rescale, you get a similar distribution, but it'll have a different scale parameter. This makes actually testing, this is actually, turns out to be kind of important for testing these distributions, because it's almost impossible to get a random sample from a set of adaptive mutations. Why? Because natural selection picks the more fit ones more often than it picks the less fit ones. And so you tend to miss a bunch of the mutations near the beginning. But instead of measuring your fitness relative to the wild type, you can do a shift out maybe a couple of mutations and measure them out of that, and then that will kind of correct for the fact that you're missing the beginning ones. So it actually is useful in that way. And for the exponential, you can see you get the exact same distribution when you shift. And this is a power analysis, and this is where Craig Beisel did this power analysis. And this has kind of saved us from, because we had pretty much thought about giving up on this particular project, because we have a little catch-22 going on. One, if extreme value theory is in play, you should expect only a small number of mutations that are adaptive. If you have a small number of mutations that are adaptive, you don't have enough data to tell whether extreme value theory is in play. And the thing that saves you is, if you're in this Weibel domain of attraction, you can get by with very few mutations and still have reasonable power. Whereas if you're over here in the Frichet domain, it's very hard to tell. So 10 mutations, which is not unreasonable to see in an experiment, a kappa of -1 has a fair amount of power. Now what we mean by power is if you're trying to detect whether the exponential distribution fits the data, or whether one of these truncated ones fits the data. And what it's basically saying, if one of those truncated ones fits the data, how likely is it that you will be able to detect that? And there's about over 70%, about a 60-some% chance that if kappa is equal to -1, you'll be able to detect it. And that's because kappa equal -1 turns out to be the uniform distribution. And uniforms look quite a bit different pattern than exponentials, and so you're able to tell the difference.
Wait, but you can run many replicas, but if you keep finding the same distribution.
Oh, if you keep finding the same mutation, yeah, you have to just basically remember that we're not counting the frequency at which we see those mutants. We're just looking at the different distinct number of mutants and seeing the pattern of those fitnesses, and see if the pattern of those fitnesses fit a distribution.
Will help you, but
right.
in a diminishing way.
In a diminishing way, yeah. Yeah, so more mutations help, gives you more power, but you can't expect to see really a ton of mutations if extreme value theory. But so that, since our data was consistent with out here, and since it has reasonable power out here, we were able to continue. We even used the phrase catch-22, and we referenced Heller's book in the paper, and it didn't get taken out, which is so it might be the only paper. I assured that the referee would make us take it out. So okay, so it turns out that what I'm showing you here is this is the exponential distribution, which you would expect these gaps. This is the average of an exponential. That's the average of a uniform. Amazingly, you get the data here. You get a kappa of -1 as your maximum likelihood estimator, and you're easily able to reject the null hypothesis. We then had a bigger data set later on in 2010. We did it again. This time, again, we were able to reject.
So what is the null hypothesis?
null hypothesis is that it follows the exponential distribution, which is the standard extreme value distribution, against the alternative that it's one of these truncated distributions fits better.
Okay, but you're saying it's whether it is or isn't.
That's if you, well, you can do it two ways. This one just does a confidence interval. It says
Yeah.
that those are, and that's probably,
But
we like that.
just as you were describing it, you say you can't test whether or not it fits.
Well, I was saying that we were worried that we wouldn't be able to tell whether or not it fits because of the low number of data, but because the power is pretty good when you're in that one domain of attraction, a small amount of data does work. We were worried that we weren't going to be able to do this, and then in the end it turns out we were, fortunately, because the data pointed to the distributions where have reasonable power for small amounts of data.
What does it mean, what kappa is? So if you...
So that's a good question. So very good question. What does kappa mean? So at this point, kappa is just a parameter that varies, that if kappa is negative, it makes the distribution truncated. Kappa minus 1 gives it a uniform. Kappa less than minus 1, it starts to actually go up. Kappa less than minus 1, it kind of comes down but in a hump. And then as you get closer to 0, it looks like an exponential. So it just,
telling you something about the biology of the
so that is the whole point of the talk. What is it telling you about the biology? So at this point we didn't know, but we came up with a different explanation, and so I'll tell you that in a bit. And at least it proposes what it might be. So here's where we were at that point. We considered this expanded view of extreme value theory, or just considered the exponential. We looked at the whole, what we call the generalized Pareto distribution. We got a better view of how it might work. We developed the statistical theory, and we were able to fit our phage data to it and decide that at least for phage, the truncated distributions are better. And that's where we were. Then we went on to the second step of adaptation. And so here's a set of mutations and their frequencies that we got after one step. Then we took the most fit background and we evolved again. And one of the things that you can certainly see is the second step of adaptation, the fitness effects are much, much smaller. And if it were exponential, you wouldn't expect that. You would expect them to have the same sort of effects each time. But if it was truncated, you would expect that.
But if you have a very small first step, then your big step.
Right, but we took it off of the biggest first step just to see, and then shifted the distribution out and then asked what was the distribution on the second step.
Oh, okay.
Okay, and now there's two ways that now one of the things that we can test is whether or not the data is consistent. Now we're looking at two steps of adaptation, whether or not the data is consistent with a distribution of mutants and then followed by a shift and using the same distribution on the shifted data, or whether you need two totally different generalized Pareto distributions. So that was our next hypothesis that we tested with our first and second step data. And what we found, and so now here's where the math, I'm going to skip over this really. This turned out to be a really nasty inference problem. And why is it a nasty inference problem? Well, there's two things going on. One, your fitness assays have noise in them. So you have a distribution of fitness effects. That's this generalized Pareto. On top of that distribution is a second level of noise, which is the measurement error. And so you have what's called a state space model. You've also got these truncated distributions which have boundaries, and these boundaries involve the parameters, which always produce nasty statistical situations. So it took us quite a bit, but so I'm going to just tell you that we tested whether or not one generalized Pareto distribution could explain both the first and second steps versus two generalized Pareto distributions, and then we added the noise factors and estimated the parameters and did the likelihood ratio. I'm going through this very fast. Isn't that a lovely likelihood surface? And so that actually, the fact that we were able to finally get an answer, and the answer is that here's the best fit distribution where these are your first steps and those are your second steps, and you force it under one generalized Pareto distribution, that's the best one. And if you do them separately and then do the likelihood ratio test, you will reject the null hypothesis, and it fits the two significantly better. You always expect it to fit better, but it's significantly better than the one step. Okay.
That's a bit disappointing.
Nope, because now we're going to get to what this means, what this might mean biologically. So here's the deal. How do you interpret these parameters? So that was exactly your question. Is there a way to interpret this kappa parameter, which seems to sort of be telling me, what kind of distribution I should expect for fitness effects of adaptive mutants? Does it have could it have a potential biological meaning? And same for tau. And what does it say about adaptive walks if you can't fit the same distribution to the first and second step? And that's what I'm hoping to at least give an idea using some ideas that Guillaume, I'm not sure I'm saying it's Guillaume Martin proposed. And the idea is the following. Let's suppose that there are a set of phenotypes that are driving this. So each one of these mutations occurs and it sweeps through the population. And let's suppose that that mutation affects a certain number of phenotypes and has a phenotypic trait value, and that trait value determines whether or not what the fitness is. So that's our theoretical framework. And let's suppose that and it turns out we can, just based on that framework, I'm going to rederive the probability distribution of the fitness effects of adaptive mutants, just on that basis alone. And the only other thing I'm going to assume is that I'm near the peak, and the peak is quadratic. And that's it. Okay? And I'm going to assume that there are a certain number of phenotypes, let's say M phenotypes, and total, and a subset of those are what are affecting each of the individual mutants. I'm going to propose a function g of x, which is my phenotype to fitness map. And I'm going to put the peak at 0, 0, 0. And of course, since I can't draw in n dimensions, I'll draw in 2. So the distance between the bottom and the fitness peak is g of 0. And this is a mapping of two phenotypes. And now I'm going to assume that around 0, around the peak, we have a quadratic, and I form that quadratic using matrix algebra, and because I'm doing this in n dimensions, but you can do it in 2 dimensions. So A is an n by n matrix, and a second derivative of a matrix of one of these quadratic functions is a matrix, and it's just basically multivariate calculus. If you remember Taylor's theorem in calculus, just think it's the same kind of idea, but we're doing it in many dimensions. Okay, and that's all I'm going to assume. And now I'm going to ask, what's the chance that the fitness exceeds a certain level? And I just go through the algebra there. And the basic idea of going through the algebra is I use this quadratic equation and ask the chance that it exceeds a certain value. S is just basically the probability. Remember, the fitnesses grow. It's just the probability of being in an ellipse. So if you're in that ellipse, your fitness is above a certain level. Okay, and so we're asked basically what's the probability of the fitness is in this. Not the fitness, the phenotypes. I said that wrong. So you take this quadratic and ask what's the chance that you get a fitness is vertical, above a certain level. And that is going to happen if the phenotypes are in this, let's see if I can, are in a cross-section of that, which would be an ellipse. Okay, and that's essentially what this probability is. And now let's assume that our phenotypes are just there at random. So the probability of being in that is basically proportional to the area of that.
There's a Fisher, is it a Fisher version of this?
Yeah, so this is basically very similar to Fisher's ideas.
But all
But he-
was kind of using those, or not
No,
really?
no, he wasn't using them at all in this particular. He's talked about Fisher's geometric model in other contexts, but not in this.
Okay.
Not in this.
So how,
Okay.
How does it relate to Fisher's geometric model?
How does this relate to geometry? So we'll show you in a minute. Basically, what it turns out is the generalized Pareto distribution can be derived from Fisher's geometric model. That's the answer. And that's what we were able to show. So effectively, you have to do a little bit more. You have to turn this ellipse into a circle by doing a transformation of variables. It's one of these tricks that you learn in calculus. And then you just go through the calculus, and voila, in the end, you end up with this generalized Pareto distribution. And so at the end, I'll take the derivative and get the density function. And so now the kappa parameter is related to the n, the number of phenotypes. And in fact, kappa is 2 over n, because that equation with kappa was a 1 over kappa. So 1 over kappa is, so essentially what it means is, what's kappa equal to 0? That's n equal to infinity. So if many phenotypes are affecting the trait, it should look exponential. If only 1 or 2 are affecting the trait, it should look like these truncated distributions. Okay? And the scale parameter is just how far you are from your fitness peak. So how far you are from the fitness peak is the tau parameter related to it, and how many dimensions are affecting the trait is the kappa parameter. So that's the answer. Kappa is basically the inverse of the number of traits divided by 2. The 2, I don't know why, it just shows up in the math.
Wait, but so in your model, there's always a maximum fitness.
That's what I'm assuming. I'm assuming that there's a maximum fitness, and then I am a small distance away so that a quadratic equation would be a good approximation. And I'm basically throwing down phenotypes, mapping them up to fitness, asking what's the distribution of the fitness effects of mutants, and it turns out it's generalized Pareto.
But isn't it always truncated? Sorry.
Yes, it is. Well, so it's always truncated for n equal 2, remember? But what about n equal 3, n equal 4? It moves away from, as n goes to infinity, the truncation point gets further and further out and eventually goes to an exponential. Yes, it's always truncated for any fixed n. It's n equal to infinity, or a large number of n, where it starts to look like an exponential.
So your kappa was, Philip kappa was 0.7, so that corresponds to 1.5 traits or something?
Yeah, about 1.5 traits. When kappa was -1, that corresponds to 2 traits.
So this, you're assuming a uniform distribution over all possible offspring.
Oh, no, of phenotype. Yeah, that each mutation affects 2 traits, takes a number of traits, and those traits are sort of chosen at random over some region.
Yeah, but I mean, in reality, you would have a type in the population, and then mutations would cause you to sample a region near to that type, right? Rather than across the whole.
Oh, well, so yeah, I'm just looking at it after. Well, it's unclear. So I think you're thinking of the genotype to phenotype map, which we're not actually doing that piece. We're just assuming that these mutations arose somehow, not modeling that piece. We're assuming that when they arise, they're adaptive. And to figure out how adaptive they are, we measure their phenotypes and map it up to fitness, and that gives me their fitness effect. And that ends up giving me the same distribution as I got when I used the extreme value distribution. So here are some examples. N equal 1, if it's just one trait, you would expect that the fitness distribution to look like that. So remember, this is the distribution of fitness effects. So you would expect it to be kind of piled up near the boundary, and you get there very quickly. N equal 2 is like a uniform. N equal 3, it's kind of coming down. N equal 4 is a straight line, and N equal 10, you can't really tell it from the
What?
exponential.
Wait, this is a plot of.
This is a plot of fitness
versus number of
versus,
diseases.
no, this versus probabilities of the, this is the distribution of, this is assuming those fitnesses were drawn from a distribution. Those adaptive fitnesses were drawn from a distribution. These are the distributions. If N equals 1, so basically what it says is, I guess the mathematical model is sample a pair of phenotypes, give that mutant that pair of phenotypes, map it up to fitness, and then ask what's the probability of the fitness of that set of what's the distribution of that set of selective coefficients.
Wait, is this the distribution of the fitness of the fittest phenotype amongst a large
No,
number?
it's the fitness effect of all of the it's the fitness effect of all mutations that exceed a certain threshold. It's a distribution of those. So think of it, you're doing this over and over again, and each time you get a different mutant with a different fitness. How would you expect those fitnesses to be? What kind of pattern would you expect those limits to follow? Would you expect them to all be piled up near a boundary? Would you expect them to be sort of like equally spaced? Would you expect them to have the biggest one quite big and then second biggest much smaller and then piling up toward tiny effects? Those are the different patterns that come out of these things. And the answer depends on how many traits are affecting it. So it'd be really interesting now to figure out if that's even true. If you could have some way to, but at least it gives some explanation. We did one generalization. It turns out, I think with stability, most think the quadratic is probably not a good model, that the surface is flatter. And so I put in a possibility of making the peak more flat than a quadratic. So I added this beta parameter, and all it does is for different values, for beta bigger than 1, I get a more flat surface than I would. And so if you add that into the model, then it turns out it depends on this flatness parameter, and the number of phenotypes will determine its distribution. And they're somewhat confounded. So now you can't tell how many dimensions if it's extra flat. So it's either extra flat or it's fewer dimensions. One of those two things could explain it. So here's our old version. There's our new version. The old version basically has two parameters, kappa and tau, which kind of are based on extreme value theory that don't have any biological meaning. The new version ties the kappa to the number of phenotypes and a flatness parameter. It ties tau to the distance to the fitness boundary.
But the flatness parameter is just.
I did that. That again is something that I'm not sure. That's just added kind of ad hoc.
Okay.
And it's just because, and I took it after we looked at after reading this paper by Weinrich and Desai. No, there's another paper by Desai, one by Weinrich, one by Desai. And they were using, and you may know better, they were trying to look at how stable mutations were, and they had these various measures of stability that you have to run these crazy programs to figure out the stability, and nobody really believes them. And so I don't know. So maybe that doesn't need to be there because it does confound things a bit. But I thought I would just, that for some phenotypes, and maybe those aren't the phenotypes you were interested in, maybe you just want attachment and the standard ones, like and lysis time and those sorts of things.
If you're in a multi-dimensional fitness space,
Yep.
okay, then you can say that individual mutations shoot you off in particular directions.
Right.
Okay, but then other mutations, like because of the shape of the genotype-phenotype space, they're likely to often be like parallel with it.
Right. So that's another way that the Fisher geometric model is used. You know, you're sitting there on the edge, and it shoots you off, and it's more likely to be a small effect mutant than a big effect mutant because it keeps you in the circle, If it's a small effect mutant, then big effect ones go outside the circle, and to stay in the circle to get more fit. That's the usual way that Fisher's geometric is used. So they don't use, so those are other types of papers, but they didn't really use it this way in those other. So this is a different way to think about it. Okay, and so again, just those are what the meaning of the parameters are. And now the last thing I can talk about is what about that first step versus second step? And here's, I'll just give you the punchline. If a single phenotype or the same set of phenotypes affect the first step and the second step, you should assume the same distribution. But if it's an entirely different set of phenotypes affecting the second step than the first step, then you get two different distributions. And it kind of goes like this. So if you can think of, if you've got the first step is determined by that slice and the second step is determined by the other slice, there's just one phenotype affecting the first and one the second. And this is just a lower dimensional version of what could happen. Then it turns out we can show that means that two distributions, you need two distributions. If it's the same distribution, then the first step brings you up to a new level, and then the second step is basically the same thing except you're at a higher level. And so if it's the same phenotypes doing the first and second, you should be consistent with one generalized Pareto distribution. So that's what it means for one generalized Pareto distribution to explain your data under this model. It means that the same set of phenotypes are affecting the first step as the second step. So that's, there's two conclusions. And the first conclusion is that to explain the distribution of fitness effects, the parameters that come out of the extreme value theory can be explained by a phenotype fitness Fisher geometric model, where the dimension is the shape parameter and the scale parameter is how far you are from the fitness peak. If you want to look at second steps versus first steps, consistent with the same distribution means the same phenotypes. Consistent with two distributions means two different sets of phenotypes. And that's it. Any questions?
So
Yes.
Within your experimental system, you should be able to test this second prediction, or can you? Because I presume they can measure things
We
like
did.
attachment. And
Well, okay, so I'll talk a little bit about that as our goal. So one of the things that's really interesting is for even really simple models of setting up these phenotypes, they don't look anything like quadratic. They have lots of different shapes. And so we have to use it, so we may be off there. But yeah, that's one of the things that we would like to start doing, is actually measuring for the sets of mutants that we have, figuring out what their attachment rate might be, what their lysis time might be, and then attempt to at least explore how the phenotype fitness map tells us something about distribution. This is just suggestive. It suggests that if you were just given a set of mutants and their fitness effect, just looking at that pattern suggests to you how many potential phenotypes might be at play. So that's kind of interesting. It's suggestive, may not be right, but at least the patterns fit the distributions. And these aren't every distribution. They're not, normal distributions don't look like this. And gamma distributions, they're all these tailed distributions. So they do tend to fit those kinds of distributions. And it could be saying something about the dimension of the phenotype fitness space. Yes?
So I don't know if there's any connection here, but colleagues of mine have done kind of bioinformatic analyses showing that more complex, or traits that have greater numbers of genes underlying them and presumably greater numbers of phenotypes underlying them
Mhm.
are much deeper in evolutionary history than traits that require just a few genes, which are very distributed on the tips of the evolutionary tree. Is your
So this
meshes
is,
that at all? Or?
that's interesting, but here I think we're kind of actually determining the evolution. So we start at the wild, I mean, we're running the evolution in real time. So we're not in this, at least in the examples, but you don't have to just apply it to those kinds of examples. So it would be interesting. So there is, that's probably true. I don't know if this model would inform that at all. It's really trying to get at how large phenotype space might possibly be. So yes, there's all kinds of more biology here. This is really a fairly simple model, and it just gives one, at least the parameters have some chance of being interpreted. That's as far as it goes, really.
So if you did know about the phenotypes of each mutation,
Yeah, you could try. You could test it. You could start to test it. It's tough. We've been working on that for a while. Also, phenotypes are noisy too, and yeah. Presumably, you could. That's exactly right. And you could at least, and even if this prediction doesn't hold, you would at least learn some things about distributions of effects and new things about adaptation, just by, it'd be worth doing regardless of what the theory sort of shows, I think, because it could point to new potential models.
Does this have any implications for the genotype-phenotype map, or?
Yeah, this is what it's just basically claiming, oh, the genotype, yeah, this is the phenotype fitness map. So the genotype phenotype map, it doesn't, that would have to overlay on top of this. So the genotype, the phenotype would, could be, at this point, it has a very simple genotype phenotype map. It just basically says that you pick the phenotypes at random, so it doesn't have a very complex view of the genotype phenotype. But those are the three steps, right? Genotype to phenotype, phenotype to fitness. So if you skip the phenotype and just go to the fitness, does it tell you something about what the phenotype might have been? That's kind of what it's trying to do.
But if a model that picks the phenotype at random does an okay job of explaining the data, doesn't that tell you something about the genotype?
Phenotype map, yeah.
It must be sort of random.
It seems like at least you're probably not being able to detect patterns. If the patterns are there, they're not strong enough to detect, but it might be you just don't have enough. This is fairly simple data, so you may need more complex data to answer that.
yeah, it might be that if your genotype phenotype map is, if it has a reduced number of dimensions, if you can only go in certain directions, it might be that you see a smaller, you think there are a few dimensions. The phenotype space has many dimensions.
Yeah, that's right. This is the phenotypes that are, the phenotype space itself could have lots of dimension, but it may only be choosing a small number.
The explorable bit.
Explorable bit might be a small number, and that's what it's actually saying in this model. Because it looks as if, and I suppose from a phage, at least Holly was not at all surprised that it was two distributions, because often what happens in adaptation is you fix one problem, and the second step is basically either fixing a new problem or just doing a little tweak on what's left over to still fix. So that's why your second steps are not as fit as your first steps.
Fixing the consequences of the first mutation.
Yeah, that's right. It could be. It could be. Because while it makes you fitter in this way, it might make you less stable, and there's lots of different ways it could go. Okay. Do I turn this off when I'm done, or do I wait for him?
You can turn off the
Maybe
box. Oh, he's here.
Oh, here he is. Don't